Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. Thursdays at 7, we'll see you there.
So it's really useful to try to figure out, how do I get myself into this desired target emotion state? So I want to feel more joyful. Maybe just thinking about happy memories isn't enough. Maybe I need to try some things and work with the system. And obviously the system can tell me if I'm getting there or not and reinforce. You have to be willing to put in a little bit of work to understand how to use the system with this equipment. But it's not too bad. It's way easier than using, obviously, an experimental system where you have like 128 electrodes or something like that. And yeah, it's pretty easy to use. And I definitely think that these will be, in households around the world before we know it.
Well, when you develop one with Pink Floyd music, let me know. I'm in.
Okay.
Hi, hello, and welcome to the Yonglu Lin School of Medicine at NUS and the Healthy Longevity webinar. Thanks for joining us. You just saw a snapshot from our last episode with Kat Agris on music and mental health. It was a bit of a departure, and I think a very interesting one. You can catch the full episode on YouTube. Tonight we have Dr. Ben Blue with us, and we'll be coming to him in a moment. And I want to remind you to use the Q&A function to send questions on Ben's presentation. Also, we have Max Unfried, a research fellow at the Health Longevity Translational Research Program, with us to moderate your questions. And even though I'm a little bit afraid to say this, let's learn a little bit more about Max.
Hi, I'm Max, a research fellow at the Center for Healthy Longevity at the National University of Singapore. I work on one question: what controls maximum lifespan and how can we change it? I'm a theoretician, and I model my day-to-day after gentleman scientists from the Victorian era. I think, I write, I drink coffee or tea, I take long strolls to ponder problems, and I meet people to discuss ideas. Come, let me show you one of my latest thoughts that I have. So what I'm really interested in is what controls maximum lifespan. And why is it that certain large animals like whales or as humans can live for centuries, whereas other short-lived animals like rodents, your mice and your rats can only live for a few years? And we know that allometric scaling laws, that lifespan roughly scales at the power of 1/4 of the mass. And what I'm particularly interested in is what are those deviations? We know this gives you a rough idea, but really, what explains this part here? What explains the difference between the actually point of the animal or the species and the theoretical allometric scaling line? Just now I was telling you that different animals of different sizes have different lifespans. And the way I've been thinking about it is, what is actually happening at a very cellular and molecular level, and trying to derive maybe those lifespan rules from there. And if you think about it, if you have a cell in your body, right? It's in this 37-degree temperature in this wet environment, and just due to basic physics, you always have thermal fluctuations, right? And even if the cell is doing nothing, right? It's just sitting there, you always have thermal kicks that does something to the cell, right? And those thermal kicks, they induce pore openings, so certain ions flow in. And for the cell, trying to stay alive, even if it does nothing else, it always has to try to pump those ions out again, right? All those intruders that go in, it has to pump them out again. It has to keep the membrane potential. And this is just when it's dormant. But of course, a normal cell does other things, right? It does things like defending against infections, right? You might have like getting some like viruses in there that like that try to do things, right? To the cell. The cell is also like, of course, like trying to reproduce and like doing all those other metabolic processes, and not only just like trying to survive. So, and all this costs energy. All this has to do that the cell has to maintain itself and creates like a basic minimum requirement for the cell. Now the issue is, if the cell wants to reproduce, right, it wants to double itself, then it also needs energy for this doubling, for reproducing, for self-replication. And in a way, this is where like you start having like certain constraints that fight with each other, and that all the energy that the cell uses for reproduction cannot be used for its maintenance. And through that, you slowly get a less maintained and maintained cell, and therefore you get damage that accumulates, which eventually leads to aging. And how those two things connect is that my current calculations show that this delta between the human and this line is mainly given to like those energetic constraints that animals that live much longer for their body mass are just better at distributing this energy accurately, and therefore are able to maintain themselves better and therefore live longer.
That's it from me. If any of this sparked any questions, big or small, please drop it in the chat. I'll be moderating the Q&A later, and I'd love to pull your questions into the discussion. You can also follow me on X at MaxUnfried, like Unfried Chicken, for spicy takes on longevity and updates on physics. Thanks. See you in the session.
Thanks a lot, Max. Very always stimulating our mind to think about what causes aging. So our guest today is Ben Blue, the CEO and co-founder of Ora Biomedical. Trained in aging biology and lab automation, he completed his PhD under Professor Matt Kaberlein, focusing on drug screening platforms using the worm C. elegans. His work bridges automation and aging biology to accelerate translational resilience-focused therapeutics. The topic tonight is Ora Biomedical, a high-throughput longevity screening as a drug discovery engine. Thanks for joining us, Ben.
Thanks for having me, Brian. I'm really excited to be here today. Thanks, everyone, for joining today. As Brian said, I'm Dr. Ben Blue, CEO of Ora Biomedical. We're an early-stage drug discovery startup founded out of the University of Washington and housed here in Seattle, where it's about 3:09 a.m. my time. So if I say anything incorrect or wrong, we'll blame it on the time zone difference. But I'd like to start by talking a little bit about why I'm excited about longevity research and what got me started here. This stems a lot from some of the early stages of my life. I was lucky enough to be raised in a 4-generation household. My great-grandmother, Clara, hopefully you can tell I'm the one in the middle. She lived to 111. She was the oldest woman in Alaska. And, and this quote is from her from a book of wisdom from centenarians. And one of the things that I always thought was really, really exciting and interesting is watching her age relative to other members of my family and either other elderly adults. She was very cogent, cognizant, and mentally aware, physically active, right up until an injury caused a precipitous decline late in life. And so early on, I was motivated to start understanding, how and why we age and potentially what we can do about it to better allow both myself, selfishly, but also others to live longer and healthier lives, much like my great-grandmother Clara did. And I do, one small thing that I like to bring up when I'm talking to folks that are in the aging field is, she was asked what's secret to long life, and she does mention don't eating cornbread for breakfast. And I think that dovetails well with about some of our thoughts around dietary restriction and potentially intermittent fasting. So from there, as Brian said, I went my background's heavily in inventing novel tools, focused on automation. We spun out Ora about 2 years ago, about 2.5 years ago now, with a focus of looking at developing new aging therapeutics. And so with this, one of the things that I think is, really important is to classify, what is aging that we're looking at? What is the reason we're going after this? And so, I think about it in the context of, my great-grandmother. It was aging allowed her to be healthy and active longer, but highlighting one of these main features here of decreased injury resilience. As we get older, we lose our ability to recover from damage and injury effectively. If I go and I join a pickup soccer game at this stage, so someone in my mid 30s, I am still taking my life into my hands, and if I get knocked down, it's going to take me a little bit to get back up again. And so there's these other hallmarks that we can look at that all lead into kind of our eventual long-term understanding of longevity, which is maximal lifespan extension and death. But I think it's important to look more proximally than that in the short-term future, and that's where we're thinking about injury and resilience. And so as someone in the space, there's two questions that I think I was thinking about as I put together this talk, and I kind of wanted to highlight as a framing narrative for what we'll be chatting about today. The first is, how do we, both as Ora Biomedical, as myself as a researcher, but us as a field of people undergoing longevity medicine investigations, how do we find new molecules that will eventually extend healthy lifespan? So that's the science half of it. And then the second half, still grounded in the science, is how do we actually take our insights from those discoveries and move them closer to the clinic in the near future? I think we can all take it for granted that doing human lifespan studies take a very, very long time. Doing clinical regulation of those take a really long time. And that's why I think you'll see Ora Biomedical, like so many other groups in the longevity medicine space, taking insights from these fundamental biology mechanisms of longevity and then applying them to more proximal cases. And so that's some of the work around injury, recovery, and repair and human resilience we'll be talking more about now. So the next point I'd like to kind of touch on is, why we think about longevity medicine as an engine for drug discovery. And historically, drug discovery has been really, really slow. A lot of it has been because we go after treating the recovery from injury, treating symptoms, treating illnesses and sickness after they've developed, rather than truly preventative medicine. We were also constrained by a lot of complex regulatory mechanisms that really drive us to develop a single drug after a single indication. So, we might have one drug that improves resiliency for a neurodegenerative disorder, another that helps improve immune function. There is some underlying mechanisms that govern both of those. And by going after longevity medicine, by treating aging as the upstream factor for multiple different indications, we can find therapeutics that are going to be much more efficient when we think about the amount of money that we need to develop them into clinical marketplaces and get them doing good to patients in clinics sooner. And so I think this is important to highlight. One lead drug into multiple indications is really one of, I think, the strongest arguments for why we as a community of people excited about longevity can be pushing those that are on the kind of more traditional pharma and biotech side into moving into listening to us and to our insights. How do we find these longevity drugs is then the next question. And so I like to throw a couple of these examples up, starting with an N of 1 experiment I've been running for about 35 years on the left-hand side of the screen. These are my student ID cards from undergraduate, graduate school, and then now recently my newest headshot at Ora. And what's interesting to me, and something that I think I like to focus on, is while the molecular mechanisms will be subtly different between me and different animal species, when we look at large phenotypic changes, myself compared to the young and old mouse, the young and old dog, I get gray hair much like these animals do. I get changes in body composition over time. I have more frailty. And even when we look at the very, very simple model animal that we'll be showing a lot of data from today, C. elegans, over on the right, I think most of us could look at the top image and the bottom image of the worm and intuitively know which one is older and which one is younger. There's these conserved aspects of molecular aging as a function of entropy, where we see disorganization and breakdown of structure over time. And that's something that I think is important to highlight when we start talking about early discoveries in model systems. A lot of them may not necessarily one-to-one translate on a molecular level, but by using insights from them and using these analogous mechanisms and conserved bits of aging, we can better understand the broad scopes of how large aspects of chemical space are affecting underlying aging biology, and then extrapolate them backwards through rigorous testing into more complex models. And so at Ora, our goal was to build a platform that would let us look at those vast quantities of chemical space that have really been unexplored. And so, while an incredible amount of very good investigative research has been done across some of the biggest candidate molecules we have as a field right now, so rapamycin, metformin, a lot of our discovery and works developing new molecules has been focused on derivatives and molecules that look just like them. And so what we built was this platform that let us start with understanding phenotypic aging as our first screening point, rather than going from a cell culture-based screen where you're looking at a molecular hallmark. We wanted to look at actual whole animals. And so this system uses lifespan of these animals as a fundamental readout, and then uses a iterative process coupled with AI drug discovery development to then iterate on hits to make ones that are much more capable across indications that we're really, really excited about. That are downstream of longevity medicine. So everything from radiation therapies to cancer prevention. And so this AI-enabled discovery engine is powered by a set of custom-built robots designed by a co-founder of ours, Jason Pitt at the University of Washington. And then my PhD research was developing our set of AI analytics that go and track the animals over time, looking at their behavior to better understand their overall lifespan, as well as metrics of health as they age. I'm going to show you a couple of videos of what this looks like in real time. So this is actually videos taken at our lab in South Seattle. And so these are different sets of plates that are undergoing experimental data collection at any given point. We have these 3D printed racks that allow us to scale and multiplex these experiments. And we put the animals on for about an hour. We let the camera go. It collects data over time for each of the individual wells on each of those plates. So there's about 144 individual wells, each one with 25 animals in it. So at scale, this means that any given flight, so set of experiments, has about 3,000 animals and can have anywhere between 20 to 50 individual interventions. And then what this data looks like, we're going to be looking at 3 different interventions. So a control, DMSO, it's a solvent that we commonly use as a control vector to dissolve the compounds in. That'll be here, and you'll see it color coordinated between a lifespan curve over time, and then 2 different treatments that we found extended lifespan. And so over the course of about 30 days, which we ran this, we found that this drug, Orb 1052, actually had sufficient lifespan extension out to where some of the animals were even alive after 30 days, which is pretty robust for these traditional experiments. Normally, these animals' population will live anywhere from about 16 to 19 days median lifespan on average under normal culture conditions. And so seeing us where we've got a median age out past 25 days is really, really exciting. And so this molecule is one of our exciting new candidates that we're moving forward on. I'll let this play again, and you can see as animals stop moving and are called dead, they receive these curves over time. Now, traditionally, these experiments are done with a unfortunate graduate student or undergrad or technician coming in every single day, looking at each of those 30 animals, poking them with a wire rod to determine if they're alive or dead. So at scale, when we're running hundreds of these novel drugs each week, it's actually a saving of hundreds of person hours per set of experiments we do when you think about the average student coming in to check those every single day. And so right now, what we've developed is this first-in-class library of longevity extending molecules. Interestingly, and I'll point this out, so this is one of our best molecules, ORB-1284, and we'll be talking a little bit more about both the other mechanisms of this later on in the talk. But we also find things that decrease lifespan. And so this is particularly interesting when we think about the overall capacity of the longevity medicine space. Right now, the excellent database DrugAge sits about 1,000 to 1,500 unique drugs that have been tested for longevity and compiled from open access publications. Right now, Ora is sitting on several times that, actually about almost an order of magnitude greater number of molecules that we've screened. I will say the caveat to that is that they're highly enriched for things that have had at least a publication around a positive effect, since so much of the literature is really focused only around reporting positive modulators of lifespan or aging rather than negative regulators. And so interestingly here in this heat map, where each box is a unique intervention, we actually found a number of molecules that are interesting to look at because they're actually deleterious to health at the tested concentrations. And so these can range from anything from unique novel compounds that no one's looked at before to even drugs that are previously FDA approved. One of the strongest negative regulators of lifespan that we found, we actually saw our first bit of data on it in about 2020, and it was the antihelminthic ivermectin, which was heavily in the news at the time. Turned out it worked great as its intended purpose as a dewormer and effectively dewormed our experiments. And so what this gets at is we can start looking at this large library, and we're seeing, in this case, this is based on some open access data that we've published. So this isn't comprehensive of the whole library, but what we can look at is what are the new and established pathways that are highly conserved. And one of the things that I think is really interesting, particularly for folks in the longevity space that have been working around rapamycin and mTOR signaling, is that if you look on average, rapamycin is produced, or the mTOR pathway, which includes rapamycin, is highly enriched for molecules that extend lifespan, with the caveat that there are even some that have a incredibly deleterious effect on lifespan, even down to as low as 50% drop in median lifespan. Another aspect that we can look at that uses the simple model system is, while we start primarily with lifespan, we can also look at other disease models. And so this is going to be a similar graph. And what you'll see here is an experiment that's run over a shorter time scale, about 4 days, looking at a model of proteotoxicity governed by one of the proteins implicated in Alzheimer's disease. And so these animals get paralyzed. And what we've actually found is a couple of synergistic reactions here where drugs plus, in this case, metformin, which was a co-screened molecule with this, worked really well in conjunction with each other. And this is a whole lot of space that, great work by folks at NUS like Brian and our other co-founder, Jan Gruber, have been investigating. And so inspired by their research, we conducted this pilot screen and we found some really exciting potential new therapeutics that we're looking at in the context of neurodegeneration. One thing I do want to highlight, and so, we built this platform that's letting us go faster than just about anyone else. But what I think is important to recognize is that the longevity field is heavily community focused and community science driven. And so one of the things that Ora and myself are committed to is not just enhancing, our treasure chest of exciting molecules and then hoarding them. We're really excited to put as much as we can out there into the open ecosystem, even as a for-profit company. And so we started the Million Molecule Challenge, an idea by my co-founder, Matt Caberline, to provide open access data of 1 million compounds. And while we're well short of that in terms of overall goal, we've still had an incredible bit of progress working with community scientist members from all over the world. A recent example of this is working with Rapamycin Longevity Labs, where we've just successfully finished the fundraising for a 600 novel mTOR modulator screen. We've already released the first half of the results. That's one of the lists you can see on the right. We'll be releasing the second half of that as soon as we finish screening, now that it's funded. And actually, I'm really excited to announce today, this is, I think, the first formal announcement of it, is through a philanthropic donation, we're launching a screen of 700 bioactive peptides that have not yet been looked at in the context of aging and longevity before. Some of these we are going to select from peptides that are of interest to the longevity community from a consumer perspective, but some of them are completely untested and unscreened. So we'll be posting that results from that and making it open access when it's available shortly. And so what this gets us at is this engine. So we can use not only lifespan, but this complex set of phenotypes in conjunction with our own data sets, publicly available data sets, to not only identify new therapeutics in chemical spaces no one's looked at before, but then also iterate on them, finding new combinations and combinatorial therapies. But this still is longevity in somewhat of a vacuum, right? How are we going to get this to a prescription someone can take? So this was an exciting bit of research that I want to spend the rest of my talk speaking about. And so I don't know if anyone here other than Brian has met me in person, but you'll know pretty quick, I'm a big old nerd, and I have been my entire life. I've always loved the idea of, what could science do to make science fiction real someday? Really excited about the future of spaceflight. And so I was thinking about this the other day, and I was noticing work by David Furman, among others, including the NASA twin study, that indicated that people under extended spaceflight conditions exhibited biomarkers of accelerated aging. And so I started to think about this in the context of what is actually causing that. And so it's microgravity and radiation. And so I had this idea that what if we took our best lifespan extending molecules and use them as therapeutic treatments for radiation exposure? So this would be looking at things like radiation damaging DNA. And so outside the context of even space medicine, this has a lot of avenues. So, if we think about DNA damage repair, that's a core hallmark of aging. We can think about it in the context of an aging therapy. We can think about it in space medicine. If we can actually improve DNA damage repair, we can also start looking at things like skin cancer prevention, even cosmetics from that angle. There's also a huge interest across national defense. And then the thing that I think is most important, and what we've found a lot of really solid data around, is looking at actually improving cancer care for patients with this. And so this is where we're focusing right now across our immediate drug development goals. And so this is asking a question, what if cancer treatment, i.e. radiation therapy, didn't have to harm the patient? And so what Ora does is we use our drug discovery engine based on longevity medicine to harness the biology of aging to target therapies that repair DNA damage and allow cells to heal from radiation injury, essentially like they're younger. And of course, targeting this at healthy cells while allowing radiation to still affect the cancer. This is a really large unmet need. All of us, I think, have had our lives touched in some capacity by someone who's undergone cancer diagnosis or cancer treatment. My father-in-law actually lost his sense of taste following radiation that successfully treated his cancer. About half of patients receive radiation therapy. 95% of them have some sort of radiation dermatitis. So this can be as mild as like a severe sunburn all the way to an open wound. Causes pain, emotional distress, can be horribly invasive, and it leads to a lower quality of life. And so what we decided to do is take our best-in-class longevity therapeutics, and we partnered with the US Air Force and Space Force, took, in this case, Orb 1284, our strongest lifespan extending molecule, developed a model of radiation resilience in our invertebrate animals, found that it worked to increase survival, found that it increased cellular survival following radiation at a degree greater than rapamycin. And then, just like we'd expect, it also increased markers of cellular senescence to a degree similar to rapamycin at 1/200th of the dose. It also, this is again highlighting actually the, looking at irradiated cells in culture, the survival rate following radiation. And then diving in deeper, what we found is that it actually works by improving DNA damage repair. So people think of DNA damage being buffered by antioxidants. You can basically stop the damage from occurring. What we found is that the cells are actually undergoing DNA damage, but then repair is occurring much quicker with these treated molecules. And so this gives us a really insight into how we can treat patients. So what we're developing is a topical formulation that will get applied to the skin of the patient, improve the skin cell's ability to resist the DNA damage from radiation, allow them to actually still benefit from the radiation therapy by allowing it to access the cancer and kill it, but keeping the healthy tissue resilient. And so this is exciting from a lot of perspectives. Codes exist for billing. So for those of you that are familiar with drug development paradigms, wanting to get insurance to be able to cover this, at least in the US system, is really, really important to a successful therapy. And the benefits to this are not just with the immediate treatment, but also long-term benefits of having a longevity medicine. Cancer patients that get this are much more susceptible to skin cancer from the region they receive radiation because of the long-term damages from it. By improving the repair, we suspect that we'll actually see not only a improvement in acute symptoms, but actually better long-term prognoses. And so this is actually some recent data looking at several of our compounds, including Orb 1284, tested in human skin tissue. And this is a model of actual solar radiation, so UV radiation. And what you can see, sunscreen as a physical barrier blocks some of it. Our vehicle lets in a large amount of radiation to the tissue. And then our two compounds here mitigate it, one to a greater degree than the actual physical barrier of sunscreen. So this is some exciting recent data that we've seen as we've translated these early discoveries in invertebrates all the way now into human tissue that I think is really, really exciting. And so this gets at a large spectrum of other indications. So we're, aging skin is really relevant to many people. So we're working across the spaces of, therapeutics that bridge everything from cancer care all the way to interest from companies that are in the commercial cosmetic space that are wanting to use some of the best-in-class aging technology in their products. And so, with that, what's been most exciting has been, taking these discoveries and moving them forward alongside a really incredible team. And so myself and Blair, our CSO, have been leading the effort here on site at Ora, developing these compounds, leading drug discovery. Matt is a co-founder of ours. His company, Optispan, that he's CEO of, is housed just next door to us. They're a clinical longevity program. And so at our site in South Seattle, you have everything from the very first bit of drug discovery all the way to people that are in clinic now experiencing the world-class longevity medicine. And then, of course, my co-founders who are on the call today, including Brian, and a set of teams that span pharma development and others. And so, right now we've had incredible support. I'd encourage anyone that's interested to reach out. Like any entrepreneur, I'm always raising money, but more importantly, I'm also looking for stakeholders and people that want to work with us across projects in better communicating the needs of longevity medicine space, or even just hearing your ideas on what to test next. So with that, I want to thank everyone for tuning in. Thank you for putting up with me at 3:30 a.m. And with that, I'll turn it over to Brian.
Thanks, Ben. I think you're the only one up at 3:30, so I think we should be thanking you for putting up with us.
This is fun.
Also, I want to just point out that I have one of those four-generation pictures too. My great-grandmother was 95 when she died, grandmother 101, and my mother's 87 now, and there's no signs of my inheritance coming soon. Let's put it that way. So, so it's really a privilege to be in a family like that. Also, two other personal points. One, as you mentioned, I'm a co-founder as well of Ora, so I'm not entirely unbiased here for the audience. But more importantly, that picture of you as an undergraduate, Ben, you look a little bit psychotic, especially since it was taken in Eugene, Oregon. I think we need to talk about your undergraduate career a little bit, maybe offline though. Is that?
Yeah, I will say Eugene is an interesting place. And that's all I will say.
Sounds good to me.
Yeah.
Really interesting talk. Let me start off with a general question for you. Because I get asked this a lot, because everybody hears about AI now and they hear about drug discovery. And how much does AI really accelerate drug discovery right now? I mean, we're doing it, a lot of people are doing it. I'm a believer, but I'm just interested in your opinion. Is it a order of magnitude? Is it twofold? Is it a myth? Where are we?
Yeah. So I'd like to frame it in terms of two areas where AI either is useful or could be useful. So one where I think we're already seeing the use case is AI as a tool for automation, for discovery, for improving the efficiency of research. I'll highlight, a common thing that we've seen is that scientific literacy challenging to wade through and understand what methods people have used, were they correct, were they appropriate, and when we're going and testing on novel compound, we want to give it the best shot on goal that it potentially could be. But the literature doesn't necessarily always report what the best concentration, solvent, yada, yada. And so we actually built some of our own AI tools that let us mine the literature at scale, use open access databases of chemical structure to give us predictions of what concentration should we test each molecule at to best actually meet those needs. And really, in that case, it's not groundbreaking science, but it's improving our efficiency. So rather than having to run, let's say, 5 doses of a molecule to give it a chance, we can run 3. We can even use it, across the spaces of helping us enrich for which libraries and molecules to go after, and those day-to-day just improvements that I'm very bullish on and very excited about. The second half of this, and I think this is where people really think about AI and drug discovery, is AI predicting new drugs and predicting, what are the things that we should be developing. And I'm also bullish on that, but I also think we need to couple it with tempered expectations. We're seeing right now some of the first AI-derived molecules being put through clinical trials, right? We have yet to see one fully in clinic, at least for multiple successful years now. And so I think the jury is still out on how well those models that were used have effectively increased our chances of getting a molecule that works really, really well. I think part of that is because so much of that training data is based on publicly available literature that is at best hard to manage, at worst incorrect. And so we end up with these, you really honestly can't throw a rock without hitting a company that says they're doing AI-based drug discovery. And so, everyone is using these open access data sets. And I think one of the things that I'm excited about that sets Ora apart is that we're building the training data for AI-derived longevity therapeutics, because we'll have ground truth longevity data around more molecules and more aspects of chemical space than anyone else in the world. We'll also be making, all of our open access subsets of that available to people, so that, not only can we use our internal training data, but those from all over the world, including people we're already working with, have new tools to train on. So I think it's really exciting. I think that the sexier side of AI drug discovery still has some major milestones to hit.
Well, maybe I'll talk about the unsexy side, because we're actually using large language models to predict drugs to affect longevity. We have a paper under review now. And I think, having gone through the process with a great postdoc in the lab, I'm a believer that we're actually discovering things that are useful, and it's at least accelerating what we test. So that's interesting. But now we're extending that to peptides, and digging through LLMs about peptides is a little bit more challenging. But I think that what excites me about your project is that these peptides are not completely, but largely unexplored space for therapeutics, and you think about peptides, GLP-1, I mean, insulin, these are some of the most important drugs on the market, and we've been sort of ignoring them and putting them in this gray zone. I think that that's going to be a revolution in the activities of different peptides and therapeutics. So I'm really excited to see what your screen does, and if it aligns with any of the predictions we're trying to make.
Yeah, it'd be, one of the things that I'd love, and we can, continue this discussion, but also anyone else at NUS or collaborators, if there's ideas of peptides that, we can solicit now, well, we have our library of 700 peptides picked out. We can still add things to it as we go forward and really spin up the screen over the next 2 weeks. So we'll happily, if anyone else here is a community member, wants to throw in ideas, my contact info will be here. Feel free to send me an email, and Brian, you and I can chat about that as well. I'd love to throw in some of your predicted molecules.
Just our initial hunting, we found about 300 peptides that had been somehow mentioned in the context of aging and longevity. So in the literature. Awesome. So we should compare notes.
Yeah.
I'm going to ask a question that maybe Max will like, and he'll probably follow up on. So one of the questions is that when you have a molecule or any intervention that extends lifespan, what's going on with the shape of the longevity curve? So you can have scaling interventions where, all the way along the survival curve, the lifespan is extended, or you can have what I call squaring interventions where more of the animals get to the maximum lifespan, but the effects on maximum lifespan are diminished or are non-existent. Is it easy to distinguish between those two things, and is one more common than the other?
Yeah, it's an interesting observation. It's an interesting question. We've actually gone through, and this was to take you guys under the hood, this was a question that I asked our ML engineer to speculate on during his interview with our team, was to use a model to look at curve shape and predict what kind of lifespan effects are actually happening under the hood, and can we classify compounds just over those kind of main categories of what we see. With some of the early screening tools that we're doing, I've been in tool development and automation, and especially even invertebrates like our humble C. elegans for a long time. I think that the most exciting work for those is moving at scale through libraries and being detail oriented in how you do the research, and then immediately translating those into in vitro assays, where the thing that I worry about with a lot of early invertebrate research is that you can go and do those dynamic observations that I think are really best suited to, say, working with a mouse or other more complex mammal population. And you can do those on C. elegans, but then you realize that, it's part of invertebrate biology that might be driving some of that specifically. So I think about it quite a bit, and it's a great question. I think in this context, we see everything from, I think I showed a lifespan data set here where we actually had a longevity intervention that increased median lifespan, but not maximal lifespan relative to control. And then a third one that increased both median and maximal lifespan. And so we can start to untangle some of the features of that based on those, and we can start to think about those as early mechanistic insights. But I really want to emphasize how quickly we go from just the rough, does it extend median lifespan, straight into human cells as our first bit of triage.
Maybe I'll ask one last question, and you emphasized the point with the graph that had the red and the blue dots, that things can shorten lifespan as well as extend lifespan, and you have lots of examples of both. I think the thing that struck me about that is seems like there are at least as many blue dots as there are red dots. And I think that may be skewed a little bit based on how you're picking things, but nonetheless, it suggests that extending lifespan and shortening lifespan are probably not that different in terms of frequency of testing drugs. Would you agree with that? And what does that mean?
Yeah. So what we've been looking at is, one of the questions we've asked is, what is good enough to tell us that a drug is worth investigating more? Our platform is the first line in the sand. And so we often set our benchmark about 30% or higher percent increase in median lifespan for what is truly an exciting novel compound. Those below that are still interesting, and often we can get health metrics out of that are really, really quite robust. I actually was talking to our other co-founder, Matt, and so I was analyzing the data from the first half of that mTOR screening project. And what we saw from that, set of mTOR modulators was kind of what you are getting at. There's a bell curve there, and it looks like a traditional bell curve. Even with the mTOR enrichment in our system, we saw maybe a slight, 5% rightward shift in the overall bell curve. And you still see things that are really, really robust in that. And you see things that are really, really deleterious, but most of it ends up being about zero. At Ora, if we think about our most exciting molecules being 30% or above, the math I did recently was about, a 3% hit rate in terms of screening. And this is largely unbiased screening.
Yeah, I mean, it's easy to extend lifespan. I think we see that in all these invertebrate models at least. And I would argue it may not be so hard in mice. Says something about whether we're optimized to live long or not. But maybe rather than delve too deeply into that question, I'll bring Max in because I'm sure he has a lot of things to ask you.
I have a lot of things to ask Max about being a gentleman scientist.
Hello from my end. We've got a bunch of questions from the audience, and those questions have priority. And one of them directly goes into your peptide discussion. And it's from an anonymous attendee who asks, peptides are often broken down quickly by the body. How is Ora using AI to identify more stable, druggable peptide sequences that can survive the transition from C. elegans to mammals?
Yeah, it's a great question. So we've got a couple of different ways that we're tackling that. So one of them is starting with a library of bioactive peptides that, starting from there as a screening process, means that about a subset of the 700 molecules that we've already picked, 40 roughly have some stage of clinical development already done. And so what that gives us is a lot of insights into molecular mechanism and translational relevance, even from that starting point. And then another set, I think there's about a couple hundred of them, but still, a minority have other preclinical research done on them in some capacity or another that we can actually draw insights from. And then the remainder of them are completely unbiased. And so these are things that we know are bioactive in one form or another, but there really isn't a lot of supporting data on any of them. And so that's going to be like our kind of ground truth new training data substrate for taking these 700 and then using them as a predictive tool. But getting at, I think, the specifics of your question, which is how are we using AI to predict those? So we've already built a scraping tool that's let us go across wide swaths of the research to help us select those 700 and also compare and contrast ones with human genomic and translational data, so we can best identify ones that also have worm orthologs, so that we can ensure that if we're getting relative insights from ones with the most mechanistic discovery done, we can also ensure that we're seeing real biology happening in the animals we're testing on, even from the get-go. And so we've actually been working with the philanthropist in question, who comes from a tech background, to help develop some of those tools. It's a great question.
Very cool. Making use of all the technologies available in your armamentarium. The next question goes.
I just want to highlight, Max, that's my favorite word is armamentarium. And this is terrible cybersecurity, but I love it so much that I even have it as part of our Wi-Fi password in our office. And no one else knows that word and talks about it enough. And Max, bless you. I just want to stop the entire conversation right now and tell you how much I love that vocabulary.
Fantastic.
Borderline oversharing, but go ahead.
That's a personal connection here, you know. Next question is from Gandhi Herianto. Could you explain how the AI-powered robotic platform specifically identifies hits from animal tracking for cancer therapies and radiotherapy optimization, and what advantages it offers over traditional in vitro screening methods?
Yeah, so another good question. So I'll separate that in two things. One is the specifics of how we actually go about those, and then one is comparing and contrasting us with in vitro work. And so, broadly, I'll start with the in vitro, is that people have been using in vitro work and molecular targeting approaches for decades now. We still see a relatively low success rate in terms of pharmaceutical development when we look at a lead into a phase 1 and then phase 1 through the rest of the clinical development. And what we've seen many times across most of pharma and biotech and drug discovery is that early molecular insights, you can find a molecule that will hit a protein or gene target or structure of interest that should have an effect. But then because of either bioavailability, off-target effects, toxicology, or impaired efficacy, penetration through a lipid bilayer, what have you, it actually fails to translate into a animal model, a complex animal model. And so what we're proposing is we go and start with in vivo at the very forefront. So while we're not working, say, in a direct human cell line to start with, we are looking at a complex animal that has multiple tissues, multiple cell types, and is it living in a complex environment with multiple behavioral and phenotypic outputs we can measure. And so what that gets us is that our true positive rate, I suspect, and I think our data is showing this as we've translated it out into human cells and seen effectiveness, is that we have a much higher initial discovery rate in terms of true positives. Rather than false positives because of that, because we're looking at whole animals from our initial discovery endpoint. To your specifics about how we're doing that. So in terms of the radiation resilience, so this was the work funded by the US Air Force and Space Force. And so what we built into our pipeline was actually a radiation setup where in the lab, we would go and irradiate the animals. Initially, in this early screen, we started with ultraviolet radiation as our proximal standpoint for other types of radiation, partially because of efficacy, partially because of also how easy it is to actually do in a lab setup without having to worry about a high energy radiation beam and staff interacting with it. And so we built that into our setup and then screened both our lifespan extenders and several unbiased subsets of compounds against it to figure out our first hits that improved DNA damage repair and radiation resilience. In terms of cancer, so we use one model of cancer in-house that has a gain-of-function mutation similar to the RAS oncogene. And so these animals develop neoplasias on their side. And we've developed a algorithm that goes and images the animals and then counts the cancerous cells that have developed on them. And so we can use that as an early screening tool for at least that one subset of cancer types.
I just add one thing, Ben, that to your point is especially when it comes to longevity, there is not a cell culture in vitro system to measure longevity. You can measure senescence or telomeres or proteostasis or whatever you want, DNA damage. None of those things encompass all that is longevity. And so I think it's particularly critical to be in an in vivo system if you're going to use that phenotype.
Yeah.
I would agree. I would almost like to follow up on that, but on the technological side, but I will do so in a few minutes. There's a question from Hui Tigo. It's a long one, so I'm gonna cut it short. But she also used to work in a lab and did like worm counting and poking them by hands. So you might have gone through like the same shared suffering on that. But
I salute your suffering.
But her question is, I was wondering if anyone has tried adapting your assay to screen for antimicrobial compounds. For example, to infect the C. elegans with infectious microbes, for example, like P. aeruginosa, and then test if the worms can be rescued using a library of existing antimicrobial compounds.
Oh, what was that person's name?
Hui Tingo.
Send me an email because we should talk. Yeah, so that's actually something we're directly thinking about. So, we have our core focus right now, which is the radiation resilience pipeline and developing our first therapeutic there. But we're also still exploring some of these options. The Pseudomonas assay, I also used to do way back in my first undergraduate research lab at Oregon. That wasn't the reason my eyes were like that, Brian, though, I will say. But in that, we've also been talking with, one of the ways that we think about our system is that it's applicable for a lot of different screening modalities. And so I was actually just recently on a call with some of the groups here in the US that run some of our national defense that are interested in how to do host-directed countermeasure development. And so we actually have been talking about developing the Pseudomonas application within our pipeline to help develop a rapid screening tool for use as novel, bioterror threats develop and new diseases arise. It's a really insightful thing that I'm really excited about pushing forward on.
There you go, going beyond aging to other areas. I've got a question about like tech development. I mean, you've developed the WormBot, right? And I think the main idea was like having a camera that you can get your death signal or like see your lifespan curves. But technology is obviously like developing, right? So where do you see like the WormBot being 5 or 10 years? Do you think about maybe whatever multiplexing of like fluorescent signals where you could then directly test drugs and get like light beams back where you know which pathway is being hit, or like going like super resolution where you could actually see like the individual cells of the worm and see how they change? You mentioned, I think you can count somewhat similar to like cancer in your worms, but how do you see like the development of this entire platform to make it even more useful for other use cases?
Yeah, and I will say, Max, when your phrase not just longevity, I will say everything is longevity, because longevity, I consider the central axis around which biology rotates. Somehow you can find a way that any biological question translates back. I know you probably think that too, but I want to point that out for the people in chat. And so to your specifics, so yeah, I'm really excited about a couple of ways that we're looking to develop that. So we actually built a prototype that does have a fluorescent channel and signal where we can look at fluorescently tagged proteins that are transgenically added to the animal, and then look at protein expression over time, look at different age-associated biomarkers with that as a signal. We don't do that as much in internally right now, because we've been focusing on the high throughput screening aspect. That's where we're currently doing most of our R&D focus. So I'm looking to bring in and integrate a couple of other robotic tools. So integrating some, plate pouring and handling robots into our pipeline that allow us to go from, a couple hundred compounds every week for lifespan analysis to a couple thousand every week, and really start to get some of those numbers even higher than that. I've always looked in awe at some of the robotics that, say, In Silico is developing for laboratory automation. I think that that's going to be some of the next big steps forward for Ora, is bringing in a couple of, honestly, robotic arms to help us accelerate our drug discovery, modernize and improve some of the ways that we use robotics in the lab. And then as we go from that into this next stage, we actually were lucky enough, we received a federal grant from the NIH, National Institutes of Health, last year to help us improve some of the status quo in C. elegans field. So we actually developed this project called Elegantry, built on the Wormbot platform, that uses a suite of environmental temperature probes, as well as a high resolution or higher resolution digital camera that lets us automate and collect data around all the little user error prone areas of common C. elegans research. So when people decide whether or not the animals are the right point to use for an experiment, this tells them these are the right larval stage, go for it. If we see anything with the experiment, we go back and it's got temperature probes across all of our incubators and Wormbots that lets us then integrate all of that environmental recording data to help us, when we publish our results, we can say, look, there wasn't any issues here. We have receipts for every single aspect of it. And so I think that's one of the things that also sets us apart, is the idea of, if anyone ever has a question about the data, we have receipts. We have actually, and maybe I should, but we haven't thrown out any single image of all of our screening that we've done collectively to date. It's all sitting on these big servers that allow us to go through and reanalyze, remine it for new health span metrics. But it also allows us to go and show another lifespan curve alongside a video anytime someone has a question.
Beautiful. I like the synergies of humans with your robotic arms and AI agents and automation. With that, I'm handing it over to Brian. Thank you, Ben.
Thanks, Max, because Max and I have to go get some ceviche. We're going to dinner. And Ben, I'm hoping you're going back to bed. But thanks for joining us. It was really exciting talk.
Oh, thank you for having me. Thank you, everyone, for listening. Really always excited to, talk to anyone at NUS, and as well, my co-founders over there. So great to see all of you. And please, if anyone in chat has other questions or wants to follow up, shoot me an email.
Thanks a lot. So I have a few announcements to make before we go. There's a joint grant call for Luminex Multiplexing Innovation Awards. Some of you have received an email about the Luminex Multiplexing Awards. This is a joint grant call from the HLTRP Stem Cell Core Facility and Thermo Fisher Scientific. Win one of up to 5 awards, offering up to 10 analyte custom kits for 40 samples with specialized core facility Luminex multiplexing support. Scan the QR code for more information. Application deadline is March 9th. We also have a healthy longevity intensive course for healthcare professionals that's happening on February 23rd to 25th, as well as an Unlock Healthy Longevity Conference, which on supplements and repurposed drugs, that'll follow the intensive course and be on February 26th and 27th, coming up soon. Our next Healthy Longevity webinar is in March 5th with Associate Professor Evandro Feifang Stavam. The topic will be Achieve Healthy Longevity from Molecular Mechanisms to Clinic and the Society. And before I leave you, here's a teaser for our next episode. We'll see you next time, and I look forward to you coming back. Thanks a lot.
Hello, my name is Sharna Jayaprakash. I am doing my final year of PhD, and my research involves identifying how learning and memory processes are affected in Alzheimer's disease as viewed through the lens of neuroinflammation.